When did it hit your mind that biologists can reverse extinction? Can you believe in these possibilities? According to a recent article, the colossal biosciences in the US are underway to bringing back woolly mammoths from extinction. The article discusses the possibilities of de extinction using a gene editing technology known as the CRISPR-Cas9 system. The technology can modify the embryos from Asian elephants with genomes resembling those of the woolly mammoths. According to Jinzhao and Kaplan, there are several benefits for the extinction compared to risk. This article proposes that numerous woolly mammoths can be released into the Arctic to restore the ecological niche with the technology. The US is offering support and funds to a team led by the geneticist George Church to conduct this research, with $15 million being the initial funds. With the advent of CRISPR-Cas9 gene editing technology, bringing back a mammoth is close to reality. However, the article discusses some of the controversies and ethical issues associated with the extinction. It can change the ecosystem for better or negatively. In order to understand this better, this project focuses on taking a historical perspective to describe the previous efforts and ethical issues for the extinction. Approximately 4,000 years ago, woolly mammoths became extinct. Based on the historical perspective, human-made factors contributed to the extinction of woolly mammoths. Biologists and scientists reviewed that cutting the rainforest, pollution, and removal of the natural habitats contributed to the extinction of many species, including mammoths. Increased human activities continue to negatively impact the environment, threatening most biological resources and organisms, affecting their survival. Natural factors such as volcanic eruption, global warming, global cooling during the ice age, and other factors such as competition for resources and inability to adapt to the environmental changes contributed to the extinction of the organisms, such as woolly mammoths. These theories try to explain why mammoths became extinct. Biologists have continued to study the process of extinction and how to reverse it, especially for endangered species. The history of the extinction started over the years ago, with a significant record being observed in 2003, during which the cloning of birds ensued as the first de-extinction strategy. To generate the proxies of extinct species, many biological authors state that de-extinction can be achieved using three major techniques, including cloning, genome editing, and selective breeding. Other biological processes that are described to be central in the de-extinction process include biotechnological processes such as comparative genomics, germ cell transmission, ancient genome assembly, synthetic DNA, and CRISPR genome editing. Second efforts for de-extinction that became successful was achieved via breeding to restore Facile peregrinus anatom, which became extinct in the 1960s. By using the eco-evolutionary relationships, biologists were capable of individuals of the organisms interbred from different subspecies to release hybridized individuals. With selective breeding becoming successful, it led to the restoration of the peregrine falcons in the United States. Genome editing and genetic engineering are central efforts to resurrect extinct anima. Genome editing involves the process of altering and modifying the DNA following insertion, deletion, or replacement of the genome of living organisms. From a historical perspective, in the 1990s, genome editing had gained importance in the de-extinction of organisms. With gene targeting relying on homologous selection, DNA constructs can be tailored and sequenced at desired locations for knockout strategies. This offered insights. The mutations could be edited to feasibly produce a new genome, and organisms that can produce dozens of genes with characteristic traits. Asian elephants should adapt to the cold condition to become targets for gene editing. According to Harvard Medical School, George Church's team used the genome editing technique CRISPR-Cas9 to produce 44 edited loci for the genome of the Asian elephant. From a historical perspective, biologists have produced cells suitable for the production of proxy mammoths for the Asian elephant genome. However, since the woolly mammoths became extinct over 1,000 years ago, it's difficult to sequence the mammoth's genome entirely. Due to the DNA degradation, it's difficult to produce paleogenomes resembling the original conformity of the woolly mammoths. Aligning ancient DNA fragments and mapping the paleogenome allows the de-extinct proxies to focus on key genotype to phenotype pathways. The de-extinction program goal is successfully to achieve proxy phenotypes. The clustered regularly interspaced short palindromic repeats CRISPR-Cas9 system has been used to perform gene editing to produce mammoth hybrids. Gene targeting for producing edited genes involve homologous recombination and conditional targeting. The extinction has been termed WOW within the field of environmental biology. There are several justifications why the de-extinction of species is good, including scientific advancements, moral obligations, and stimulation of social interest to drive the dialogue of the selection of species. Firstly, de-extinction promotes new scientific inventions. From a historical perspective, de-extinction attempts were previously conducted using cloning and species breeding. However, in the event of new technology, these attempts offered new information on the possibilities of using the CRISPR-Cas9 technology to carry out the de-extinction and restoration of species, such as woolly mammoths. Bringing these species back in life will offer insights into how the world works, especially evolution and natural resources. Conducting de-extinction processes allows for conserving threatened ecosystems by restoring ecological niches. Restoring the extinct species allows the world to experience the past world. It's also a measure for justice on our planet to preserve more species. De-extinction involves sophisticated processes which are highly expensive and time-consuming. This will affect the conservation efforts since hundreds of living species become threatened. Conservation plans for the hybrids may become complex since they will require someone to manage the species to prevent the extinction of more species. This could lead to the rise of unforeseen consequences, such as cloning dogs or cats, and humans may be the next targets. The extinction of species could cause changes in the environment and ecobiomes. The extinction may increase threats from new diseases that may cause humans health challenges. From a historical perspective, knowledge, understanding evolution, the development of organisms, heredity, and genetics becoming crucial in biological research. It's clear that extinction is associated with the death of species million years ago. With the advent of technology in the 1960s, such as cloning, species breeding and selection, cloning, and genome editing, have proved efforts from biologists to conduct de-extinction processes. However, gene addition has become important in the de-extinction of woolly mammoths using the cross-living Asian elephant genome. Biologists term it the best approach for resurrecting animals. While it's also considered unethical, the question about the moral value in the current decades continues to deter the de-extinction process for mammoths. Thank you for listening.